So the spot you never want to have is the last lecture of the day in the B room. But here we are, and I'm honored that you're here. So let me walk you through a little story here about ethanolic extractions of CBD. So clearly, there's a huge number of home gardens, or one of the areas where I got my biggest education on cannabis. Those home gardens were actually home forests of cannabis trees. And as a result of all this legalization, a lot of people are making stuff at home. And so one of the things that I've always, being an herbalist first and then getting into cannabis, so for me, cannabis is yet another medicinal plant, a special one, but another one. I've been making tinctures forever. And the medical scientist side of me was like, well, what's in these things? So let's start with this story. So back in the days when I was in between University of Exeter in the UK and University of North Carolina, I was working on echinacea, and I drilled down really hard for 4 years on echinacea. And one of the things I was doing, I kept extracting a bunch of different echinacea angustifolia, echinacea purpurea, echinacea pallida, like all these different species that are out there in the market. And by the way, most of it is purpurea. And then some research came out just about the time I was starting my dissertation work on acrylamides or alkylamides. You'll hear both pronunciations, binding to CB2. So suddenly echinacea was like, a cannabinoid or a phytocannabinoid. You could argue that, but there was a compound in there, or actually several compounds, these compounds that would bind CB2. I once had the owner of an herb company come up to me and say, hey, if I take echinacea, is it going to keep me from getting high? No, don't worry, that's not going to happen. But so I started looking at these compounds, and like I said, I drilled down pretty heavily on them. And you can't see the way they're shaped now, but if you saw Machulam's lecture today when he was talking about all these amides, you can see that tail can fold and form kind of a partial ring. It's not really a ring, but it's an open ring, right? And so these bind CB2 rather well. And so one day I'm in the lab and I'm tincturing yet some more echinacea so I can run mass spec analysis on it and then put it in biological assays. And I tincture it, I go home, I go to bed, and I wake up in the middle of the night and I go, oh my God, I just missed an opportunity. Because there's always been this discussion in the industry, the medicinal plant industry, is how long do you have to macerate or steep plant material in alcohol before it's ready? And the traditional wisdom has been, 2 weeks is good and 4 weeks is even better. And I used to have a close friend who ran an herb company, would say, I do stuff that's 3 months and it's even better than the 1 month, and I've done 6 months and it just rocks. And so I've always been a little skeptical. And if you ask any chemist, and I know there's probably some chemists in the room right now, they're rolling their eyes right now because they already know better, right? So I woke up the next morning, went right into the lab, and I took a sample of this tincture that I had planned on tincturing for 2 weeks. Okay, so I got it at about 24 hours time point. And this publication is what came out of it. And this data was buried in a bunch of mass spec data, so nobody really saw this data. I forget which figure it was, but it was well into the manuscript. And most people, their eyes roll back by the time they get to table 2, and they're, in figure 3, and they're done. But you can see here, day 2, in other words, 24 hours later, you can see that the relative concentration of dodecatetraene, this alkylamide, is really kind of peaking. And you look at day 7, and there's really no difference. And you look at day 14, 2 weeks, which is what we're supposed to do in the industry. And by the way, in the industry, you've got barrels and barrels in all these herb shops, all these herb production facilities filling floor space because it's sitting there for weeks, right? And then so I was like, well, and let's make sure we go 21 days so that we can show that there's really no difference. Like I said, the chemists in the room are going, yeah, what's the news here? But the herbalists in the room were blown away, right? Or not in this room, or maybe you are already, but when I showed you the data I'm about to do, you're going to be really quite surprised. So I went to the literature and I said to myself, can I find anything better than 24 hours? And found a couple of studies. Now, they weren't on cannabis, but they're pulling compounds that have some lipophilicity to them. In this case, we're looking at carnosol and carnosic acid. And in ethanol at 70% versus ethanol at 100%. And then they got a methanol in there. I typically ignore that because we're not going to use that to make tinctures, right? Hopefully. So you see that really at 3 hours, there's not a lot of difference between 6 hours, but 6 hours it really kind of peaks, and there's not really much difference between 6 hours and 10 hours. So now I'm going, okay, so we've got something close to 3 hours, and we've got something close to 6 hours. So how many hours do we really need? So here's another study, and again, 3 hours in this particular case, or carnosic acid, there's no difference. So then I went to the cannabis literature, and I looked at some literature, and there are some tincture research out there, but here this one actually is really new. I think this is this is the 2008 one. They looked at 20%, 40%, and 80% ethanol. And by the way, I think most people, when they're tincturing with ethanol, they're usually using 100%. And that's what we did, and I'll tell you why in a second. But if you really want a full spectrum extract, you're probably going to want to use lower than 100%. Now, what will it cost you? It'll cost you some small percentage of THC or THCA because of their lipophilicity, but you're also going to get more flavonoids that way. You're probably going to get more terpenes that way. 70 to 80% tends to be the sweet spot with a lot of different compounds, especially polyphenols. We see that here for the most part in terms of distribution of THC and THCA. So then I said, let's take this to cannabis and check it out. So here's what we did. So we took a batch of cannabis, and this is hemp, okay, so cannabis sativa. The chemotype is CBD, right? And we used a 400 micron sieve, so kind of a coarse grind, and homogenized that. And when we got done with homogenizing, we homogenized it some more, because I knew that the whole thing had to depend on that starting biomass. It had to be mixed really, really well. So we mixed it and mixed it and mixed it. Then we took 100 gram samples, collected, and added 500 grams of 95% ethanol to that. So that's going to steep. So the maceration treatment started, but then we also had an arm, and I'm realizing now I have that arm in the wrong place, but we also took and did some percolation. I'm going to show you that data in a second, but I really want to focus on the maceration right now. So those of you who don't know percolation, I'll explain that process in a second, but I wanted to try two different traditional home-used techniques, the maceration and the percolation. And then we went through a couple more steps. We collected the Marc. The Marc, if you're not familiar with herbal medicine, the Marc is the plant material, the biomass. And then you would press that Marc, right? And then you'd get the press from the Marc, and you'd also get the pour off, the liquor that you poured off the tincture. And I'll show you these steps if this doesn't make sense just yet. And so what we did is we did independent duplicate extracts, and then we took triplicate samples from each of those duplicates, and then we did triplicate analysis of the triplicate samples. So trying to really stick with some good statistics, because I knew that the herb industry would never believe me. So there's the cannabis, and this is the starting biomass. And like I said, we mixed and mixed and mixed. And here's where we ended up. So it was about a 8.5%. Your average is 8.84% CBD. And you can see our p-value is pretty strong there. And then we ran it through the sieve, a 4,000 micron sieve. And so we had consistency. And then to really mimic home extractions, we took ball jars, which is usually what herbalists use, and I've used forever. And fascinatingly, my house used to be so full of ball jars. And then when I partnered with a lovely woman, I kind of let go of the ball jars, and now my house is full of jars again because she cans. I'm just like, those jars are menacing me in the dishwasher because I'm the guy that deals with the dishwasher, right? So then after we tinctured, we poured off the liquor, or often called the menstruum in herbal medicine. We poured that off, and then we took the Marc, which is wrapped in the cheesecloth there, and we pressed that. And we bought a press that was easily available. So usually you have these high-ton, like 5-ton presses that you use that are like car jack kind of things. Well, we just bought this wine press off of Amazon. I think it was $74, so that we had something that was really readily available and wasn't super expensive. And, you know, actually, it did okay. I mean, there's presses that'll do much better, but it did okay. So we pressed the Marc, and you'll see on the left side of the screen, that's what we poured off from the ball jar, okay? And on the right side of the screen is now the remaining liquid that was in the biomass that got pressed out. And there's a molecule we were looking for, cannabidiol. Now this cannabis was decarboxylated. And so here's what we got. At 3 minutes versus 1 hour versus 2 weeks. So in 1 hour, this extraction was done. And like I said, the chemist in here going, yeah, are you going to tell me something I don't know yet? But at 3 minutes, we had 94% of the 1 hour. So it always made me wonder, because this quick wash technique that you hear about, right, cold wash, the cold wash or a quick wash, you know, that's faster than 3 minutes. So I would guess, but I'd like to do this study, I would guess that they're probably getting pretty high yield. And then if you compare the drain versus the press CBD, and this is the problem that people have at home, pressing, right? Because what are you going to do? I mean, some people take the cheesecloth, the plant materials wrapped in the cheesecloth, and they just turn it and squeeze it, right? That's one way to do it. And you want to do that because look at the amount of CBD we got from the press. Although it was much lower volume, the amount that we got was actually pretty significant. So you always want to press, and it's not hard to do. And then we looked at the Marc. So this is the Marc after it's been pressed. Now, if you looked at this, you would argue that it looks to me like that 2-week Marc has got less CBD in it, and therefore it probably should have had a higher yield. But it actually didn't, according to the data that we have. So did we lose some CBD somewhere? I don't know. But I'm still scratching my head on this one. And actually, I don't think I don't have the stat bars in here, but I don't think this was statistically significant in terms of the 1.16 to the 0.98. So that also may be a factor here. Okay, now let's turn to the percolation. Okay, so now what have we learned? The one thing that I really want you to get is there might be a reason to run a tincture for longer than an hour, or even, I'm guessing, half an hour is enough, but that's another study. There might be a reason to do that, but it's not chemistry, okay? So people get into the moon cycles, and that, so yeah, great. If that's what you're into, maybe that's the way to do it, right? For your medicine, for the medicine you want to give to people. But for chemistry, you don't need 2 weeks, you don't need a month. you just really need a few minutes. So now how do you make a percolation cone? So percolation cones, in old pharmacies, you can still find them out back in the back shelf, hidden with dust all over them because they haven't been moved in like 40 years. But old pharmacies, this is one of the ways that we made medicine in the United States, that pharmacists made medicine in the United States for decades and decades, is they would get a big percolation cone, and by big, you know, they were this size, the ones that I know of, and they're a cone, right? And so the trick here is the liquid flow through that cone, because you've got moving liquid, which is one of the things you want. And that's something I should tell you about maceration. Every one of those macerations were stirred vigorously for 1 minute, and then the timing started. So in the same sort of way, we have movement here, and movement is important. One of the things I was talking about yesterday with extractions is, you know, there's really 4 crucial factors. One is solvent, one is temperature, one is pressure, and one may be agitation, right? So this is a sort of agitation, a movement. So how do you make a percolation cone out of a Perrier bottle? Well, you score the top of it, and herbalists have been doing this forever. You score the top of it, and now you have an opening at the top. And you hang on to your cap, the cap that you would use to close it, and that becomes a stopcock. That becomes a way of controlling your rate, okay? And so we ran that percolation, and there's been an age-old argument between herbalists about what's better, maceration or percolation. The thing about a percolation is it only takes usually a couple of hours. So a couple of hours and it's done. And if you're doing percolation right, if you go and you read the United States Pharmacopoeia from way back, when that's the way they were making medicine, they tell you to take the biomass and just really moisten it. It's not in liquid, it's just moistened with ethanol. And so if you think about that, one of the things that it does is it breaks down the cell walls. So once you do that, then with that moist stuff, then you pack the cone. And that pack is key. If it's too loose, it's just going to run, the liquid's just going to run through. And if it's too tight, the liquid's not going to run. So people that do this for a living get really good at packing these things. So here's what we got, and we're comparing it to the 2-week comparison here. So liquor recovery was about 67, that's a total liquid that we got, right? So the liquor recovery is about 67%. In the first extraction, about 72%. In the next extraction, in the perk, it was higher all around, 76 and 74. Keep in mind that we actually tried to press the Marc that came from the perk. We got 0 milliliters from it because it was already so dry. So this is another advantage. Not only do you not need a press, but you can just, you've collected all the liquor that you're going to possibly get from a percolation cone. And then if we look at CBD recovery, and that's the darker gray there that you want to look at, the Perk 1, we got about 80%, 79.2, and Perk 2, we got about 81%, as compared to about 61% versus 65%. Now keep in mind, this is not 61% CBD, this is 61% recovery of the CBD, right? So we started with about 8.8 milligrams, we got 61% of that. And then there's your graph. And I compared it here on the graph, a 1 hour versus the percolation. And you can see it's really significantly higher. I mean, you got about 15% more CBD in that percolation. Shorter time period, doesn't take up a bunch of space. So future directions for this, one of the things I'd really like to do is I'd like to do a quick wash comparison to a short maceration just to see what's going on there. I'd also like to experiment with percent ethanol. And I say that not so much because I'm interested in the cannabinoid yield, although I am. I'm more interested in being able to look at the polyphenols and comparing the polyphenol content at 100%, which is what most people are using for their cannabinoid extracts, versus like 70 or 80%. And then I'd like to do some studies with hot ethanol, with cold ethanol, and then room temperature ethanol in both a percolation method and a maceration method. And that's it. Ethan.
Kevin. A couple of quickies.
It was quick.
I didn't gather what the temperature was of the ethanol you were using.
Room temperature.
Okay.
Yeah.
So say you were using fresh material rather than dried. Wouldn't it make sense then to use the high-proof ethanol?
Oh, absolutely. So, and I thought about showing some data like that from some other work I've done. Yeah, if you're using fresh plant material, so let's talk this through for a second. So you notice that we did a 1 to 5. This is traditional dry plant, right? 1 to 5. And some people now are doing 1 to 3s just to make things stronger. But so 1 part is plant material, 5 parts is the solvent, the menstruum. When you use water, depending on what part of the plant you are, you've probably got somewhere between 85 to 95% water content. So we say in the industry that fresh plants should be 1 to 2, but if you really do the math there, it's really about a 1 to 11. So dry plant ends up being chemically stronger. There may be reasons, for example, enzymatic action in the drying process that breaks down compounds for doing a fresh plant versus a dry plant. But fresh plant, even though it's 1 to 2, with that 90% water content, right, is not really 1 to 2. More like 1 to 10, 1 to 11. Stupefied, I see. It was amazing.
Will you just tell us what that percolation process looks like as you run it, like you're pouring ethanol? I just can't visualize it. I've never done it.
Yeah, sure. So if it's, let's go back to what the percolation cone looks like. So if you're doing this right, and doesn't have to be done this way, but if you're doing this right, with the old percolation cones, they were really tall so that you could pour all the solvent or menstruum in there. And so you were done, you could walk away. Now, with smaller bottles like Perrier, you can't necessarily do that because you don't necessarily, it depends on how full you fill the bottle with the plant material. You can't necessarily do that because if it's too full, you're just gonna have to stand there and keep feeding it, right? But if you get a proper percolation cone, and they're hard to find and they're really expensive, you can just pour it in and be done.
Thank you so much for sharing this information because I feel like you were taking thoughts right out of my head. So
1 second. Hey,
Yep.
Can you turn that up? I can't hear, really. And part of that is my age, sorry, but.
I don't know. Is that better? Yeah, can you hear me now?
Yeah.
Okay, great. Thanks. So I really appreciate your talk. Thank you so much. I feel like you're pulling thoughts right out of my head. So I've said for a long time, I feel like percolations are a missing link in the industry. So I'm curious if you did other tests or you did double washes, because in traditional percolation, sometimes you do a second wash through. And then I'd be curious the other things that you tested for in the tincture, like what other compounds you're looking for, et cetera.
You're bringing up a great point. So, so I ran a production facility for a while, CO2 extraction. And we definitely looked at our Marc to see what was left, the CBD sitting in the Marc. And you know, one of the things we found was that we were only getting about half of the CBD with the CO2 extraction that we were using. This is supercritical fluid extraction, right? CO2. And so we started running again. Or we take it and we do it with ethanol. And we yield more CBD, right? So that's an option. Now, in this case, according to this data and this model, it looks like there's not much left. It looks like there's, you know, less than 20% of CBD left at the very most. And so, yeah, you could run it again and probably get more. but I'm not sure how much more you would get.
Did you test for other compounds as well besides?
No, so
No?
that's
Okay.
something I meant to say when I started this very quick lecture. We didn't have any funding, so this was all out of pocket, and we did it as cheaply as possible. But I insisted on triplicate samples of triplicate samples. So.
Cannabinoids that you want to keep when working through this process?
Sure. So it's really well. So temperature. Let's start from the 30,000 foot view level. It's really known that heat. It's well known that heat is a factor that can pull out more yield in terms of most compounds that you're looking for. Having said that, you can also use too much heat and destroy stuff. Flavonoids polyphenols start to break down around 140 to 160, depending on the polyphenol. Terpenes, as you know, can be actually much less than that. So if you look at hot water versus cold water extractions, for example, marshmallow root, right? And I'm not talking about Kraft marshmallows that you throw on a yucky milk chocolate. At least use dark chocolate. Graham cracker, and I'm talking about actual Althea officinalis. Cold extractions will pull out the polysaccharides, so it's believed much better than a warm extraction. And if it's a hot extraction, you've almost destroyed the polysaccharides. So for gut relief, the way people use it, it really needs to be a cold extraction, or at least that's the dogma in the industry. I'd like to do that study too. So it just depends on what you're after, right? Hot versus cold. And I'm going to throw this out there because this is something that I don't believe, but the industry believes. And that is that if we use cold ethanol, we avoid chlorophyll. And chlorophyll gives it this acrid taste. Well, if you can buy chlorophyll, liquid chlorophyll in the health food store. Have you ever tasted that stuff? It is not acrid. And so, cannabinoids are acrid. So why are we so afraid of chlorophyll? I don't get it. And if somebody can explain that to me after, that would be great. But I don't believe that cold ethanol is effective at reducing chlorophyll. Or maybe it is, but, well, let me rephrase that. It may be effective at reducing ethanol, at reducing chlorophyll. Cold ethanol may be effective at reducing ethanol, but chlorophyll is not a bad thing. That's what I don't believe. So heat, it just depends on what you're doing. Yep. And I'm rambling, and it's time, and I'm going to call it quits. Thank you.